Hi, this is Miss Litton, and this is a lovely lunchtime review with some excellent honors biologists. Say hi.
Hi.
And this is Chapter 4, Cell Membranes. So like I said just a few minutes earlier, that you're going to want to know all the parts of a cell membrane and what each does. So let's start by dissecting that. What's the most prevalent component of a cell membrane? Phospholipids. And phospholipids create a what? The B, I would
Bilayer?
Yeah, a layer, or barrier is what I'm after, a barrier. So it keeps some things out and keeps some things in. Okay, so your phospholipids create a barrier. That barrier, we're just doing honors bio today. Sorry. So it creates a barrier. Now, when you look at the structure of a phospholipid, it's amphipathic. I don't know if I taught you that word, but it has one side that is hydro what? Philic, and one side that is hydrophobic. Okay, so it has a hydrophilic region and a hydrophobic. Where is the hydrophobic region located, and what are they called? So take a look right here at the cell membrane. Okay, these are the phospholipids. This is the phosphate group, and these are the fatty acid chains. The fatty acid chains are hydrophobic, and they're in the interior of the membrane. The phosphates are hydrophilic, and they're on the edges, to the outer edge and to the interior of the cell, the inside of the cell. Okay, what is a second component of a cell membrane? Proteins. Good. And what do we use proteins for? Lots of purposes, right? And we'll go through all those purposes that a protein is used for. What's a third component of the cell membrane? There are four components. Phospholipids, proteins. What's another one? Steroids. Okay. So in particular, right, the steroids in there are for, you know what they're for? What are the steroids here for? Keep it together, the fluidity, right? So it holds it together if it's too hot and prevents it from freezing if it's cold. Now, do all cells have steroids in them, like cholesterol? It is cholesterol, but do all cells have it? Do you know who doesn't? Plant cells. Plant cells do not have cholesterol, but what do plant cells have going around the outside? Cell walls. Perfect. Okay, so we have phospholipids who have hydrophilic and hydrophobic regions, hydrophobic to the very center of the membrane. We have proteins, and we have all the things that proteins can do, and we'll review that. And then we have cholesterol, which helps with fluidity. What's the fourth component? Carbohydrate side chains, the glycocalyx. So those help with cell recognition and communication. And you have glycolipids, which are carbohydrate side chains attached to a lipid, and you have glycoproteins, which are carbohydrate side chains attached to a proteins, but they are made out of sugars. Yes?
Isn't the glycoprotein the only one that does cell recognition?
Yes, but you have other ones used for communication. All right, so those are the components. Now that we know the structure of the membrane, let's focus on the protein function. So let's go through the protein function. Let's do it together. They can be what? Channels. They can be carriers, cell recognition, receptors, enzymatic, and junctions. Let's go over that again because you were struggling. All the functions of proteins. They can be channels, carriers, cell recognition, receptors, enzymatic, and junctions. Perfect. Okay, then let's talk about, oh yeah, you can.
Give a review.
Honors bio right now, yeah. Then you have the ways to cross a membrane. You remember the ways to cross a membrane? Through the phospholipid bilayer, but not if you are large or charged. You can use a channel or a carrier or whole membrane, endo and exocytosis. Okay, so let's review that again. 3 ways to cross. Through the phospholipid bilayer. Do it with me, please, so you remember. through the phospholipid bilayer, but not if you are large or charged. You can use a channel or a carrier or whole membrane doing endo and exocytosis. Okay? Next big topic that you want to know, is what are the two types of transport, and we talked about passive and what? Active. Okay? Passive and active transport. If it's passive, what? Doesn't require any energy. Okay? So you're just going with the gradient. You're going from a higher concentration to a lower concentration. And if it's active, it does require energy, right? It does require energy. Okay, just a minute. Okay, so then the next thing I want to review is what is the difference between diffusion, facilitated diffusion, and osmosis. Can you differentiate those three things? Diffusion, facilitated diffusion, and osmosis. All right, so diffusion is just going from a higher concentration to a lower concentration. You put perfume on, that's going to happen, right? That's just diffusion, high to low. Facilitated diffusion is using a what? Protein to do diffusion. Okay, like you see in the second and third example over. And that's because you are probably what? Charged. If you're large, you can't get through because you're just large. Okay? But if you're charged, you're probably having to use a protein because the center of the membrane is hydrophobic. Good. Okay? And then osmosis is what? There's three things for osmosis. It's the diffusion of water across a semi-permeable membrane. Diffusion of water across a semi-permeable membrane. So it's water going from a higher concentration to a lower concentration through a membrane. Diffusion of water across a semi-permeable membrane. And when we have that, then you're talking about things like hypertonic, what? Hypotonic and isotonic. Which means there's some sort of difference across a membrane, except for isotonic is same, right? But there's a difference across the membrane. You have a membrane barrier, and there's different concentrations of water on either side of that membrane, so water's going to move. All right? Now, just make sure it didn't pause. I can't hit that. Okay. All right. Then when we also talk about active transport could just be the use of a protein like the sodium potassium pump, concentrating sodium to the outside of a cell and potassium to the inside of the cell. But this is also active transport, doing whole membrane, right? Because that's going to take energy to move the cytoskeleton, to engulf something, or to get rid of something. You all right so far? Mm-hmm. Okay, so now let's move into tonicity and hypo, isotonic, all of that. Okay? So when you evaluate two solutions separated by a membrane, if you say one is left, the other one has to be right. Okay, that's all that it is. So if you identify, you have to look at them in comparison, the two solutions to each other, to decide if one is hypertonic, and if that one is hypertonic, then by default the other one has to be what? Hypotonic. Okay? And remember we said water must flow from the hypo. Okay? So when I look at these two sides, side A and side B, which one has more water? A. Side A has more water. Okay? So it is hypotonic. Okay? Side B has less water, so it is hypertonic. And the way I remember that is, what is it hyper with? It's hyper with, in this case, with a lot of what? Sugar. Okay? So it has high solute and low what? Solvent. So when you say something is hypertonic, that means it has a lot of solute in it. You all right with that? And then we know water must flow from the hypo. Water is just going to go from a higher concentration here to a lower concentration here. And as a result, the water level on side A will go down, and the water level on side B will go up. And then we can just analyze different scenarios, right? When we talked about pickles used to be cucumbers, you're holding a cucumber right there. So hold that up right there. Okay? What do we need to put that cucumber chip in order to make it a pickle so she can put it on her cheeseburger? We need to put it in a what? Hypertonic, so the water will come out of the cucumber chip and go into that solution, and it will shrink down into pickle size. Does that make sense? Dice up some cucumbers and make your own pickles. Okay? And we looked at this scenario. They have stuff on both sides, but where is there more water? Where's the more water side? If this is side A and this is side B, where is there more water? Yeah, higher water here on side A, right? Because these purple things are something else, like salt, sugar, something, right? So side B is referred to as hypertonic. Side A is hypotonic, meaning more water, low other things. Water must flow from the hypo. So water will move this way. And then we could talk about what's going to happen with different cells if I make 3 solutions here. I make a hypertonic solution, I make an isotonic solution, and I make a hypotonic solution. 3 different solutions. And here is my red blood cell. Try again. There we go. Okay, what is going to happen to this red blood cell if I put it in a hypertonic, an isotonic, or a hypotonic solution? Can you predict what would happen if I put that red blood cell in a hypertonic solution? What's going to happen? It's going to shrivel up. You're absolutely right. It's going to shrivel because the water is going to leave it. In an isotonic solution, what's going to happen? Stay the same. And if I put it in a hypotonic solution, what's going to happen? It's going to swell because water is going to move into it. Does that make sense? Water moves in. Same amount going in is going out here, and water came out of this one. Got it? Questions you want to ask me? All right. Then that leads us to our last little bit. Whole membrane. So there's exocytosis and endocytosis. Endocytosis coming in, exocytosis going out. There were 3 types of endocytosis. See if you can come up with the 3 types of endocytosis. Not out loud, just try to type them or come up with them in your head. Okay, so what are the 3 types? Start with the one I'm doing right now with my smoothie. Penocytosis, what is that known as? Cell drinking, penocytosis. What's the next one? Phagocytosis, which is cell eating. And what was the third one? Receptor-mediated endocytosis. Okay, so this right here would be phagocytosis, like a white blood cell could be engulfing a bacterium. Penocytosis, you're engulfing a fluid with something dissolved into it. And then receptor-mediated endocytosis, it's binding to receptors before it is engulfed. All right, questions or concerns on that? You want to do the practice ones in here, or do you want to try to take a practice quiz? Okay, so study hard and have a piece of toast if you get hungry. Okay, bye-bye now. You're super smart.